Hello, everyone that's joined us today. My name's Nola D'Souza. I'm the coordinator for the Australian Fungicide Resistance Extension Network. We're known as AFREN, and we're a network of plant pathologists, fungicide resistance experts, and communication and extension providers. And we've been established to develop and deliver fungicide resistance resources for grain growers and for their advisors across Australia. So welcome to you all. This is the first of a number of webinars that we have coming from AFREN over July and August. So thank you for attending our first webinar. Firstly, today we have quite a few registrants. So I'd just like to get you all to look at the bottom of your screen. There is a Q&A button there. I'd like you to use that Q&A button if you have any questions. And at the end of the presentation from Stephen, we will be able to ask those for you. Just to continually improve our resources and our extension, we need to get some feedback from everybody that we engage with. So we'd really appreciate it if at the end of the webinar, you could do a short survey for us. It should take less than 10 minutes, and that type of feedback for us is really invaluable for helping to direct us to provide what it is that you want to hear from us. So after all that housekeeping, I'd like to say welcome, first of all, and introduce you to Dr. Steve Simpfendorfer. Stephen has been a serial pathologist with the New South Wales Department of Primary Industries and Regional Development. He's based in Tamworth in northern New South Wales, and he's been there for the last 25 years. His passion is for diagnostics and disease surveillance and communication of integrated disease management strategies to support growers and their agronomists to do these things better. He reckons he's been around for so long that he's arguably seen it all throughout his career, but he does still find effective communication an ever-evolving challenge. But I'm sure he won't disappoint you today. Thanks, Stephen. Over to you.
Thanks, Nola. So, yes, Nola mentioned this is first in a series. So we thought with this one we'd take a step back and start right at the start of getting identification correct. Really focus on cereals, which is my main thing, and just trying to put some stuff out there to challenge. I think we reach for the drum sometimes too quick. So, yeah, we hear this all the time, fungicides, they're cheap insurance. So put them on, only cost, if you use some of the cheap ones, $2 per hectare. We always forget the application cost. So I might have seen most diseases or other things in my life, but I keep hearing new things about fungicides every year. There's been a couple of great ones this year. They've almost got a mystical quality amongst some agronomists and growers of what they can actually do. They don't restore green leaf area, so once you have a disease or another thing in there, you can't get the green leaf area back with the fungicide. They only protect the leaves out at the time of application. They don't move into new leaves, and they don't actually increase yield. They just protect yield potential. That's if it's actually a disease that's causing the problem in the first place. So what's the problem with approach where we just think, well, what are we going to spray? What fungicide are we going to put on all the time? We really run into fungicide resistance issues. And how do we get there? Basically, within any pathogen population, fungal pathogen population, you've got a portion of the population that's sensitive, usually most of it, and then you'll just, through mutations, naturally out there, you'll have resistant individuals, the black ones in this particular situation. So when you apply your fungicide, you kill off most of that population. Some get missed, which are sensitive, survive, but you're selecting over time for more of that population to actually have the mutation and be resistant to the fungicide. So the more applications you do of the same active, you're really selecting for that and running into problems. If you look at this, it's evolved over time. So we call this the rainbow of fungicide resistance now. There's a range of different pathogens there, whether they've had a lab detection, so we know that it's got the mutation, which is the first step to resistance to a fungicide, whether it's actually got reduced sensitivity, so in a lab situation, put in agar plates, increasing amounts of that particular fungicide active, it's still growing well, or whether we've actually seen spray failure resistance out there in the population in the field. And that's usually when the proportion of that population gets above 50% or more. That's when we start getting spray failures. So this is not a pretty picture. It might have nice colours, but this is not good for industry, and it's changed massively. If you look at this map 2 years ago, it's changed massively in that time as well. We are picking up more we look and the more we stick with the approaches we're using. The classic net blotches. So there's things around there, spot form net blotch, two forms on the left there, clear yellow margin from the toxin production, necrotroph, and then you've got the net form net blotch. And you might have seen a press release come out recently. We've now got, on the York Peninsula, South Australia, we've got triple resistance, okay? Triple mutants. So it's not X-Men. This is not a good thing. So we've got resistance to the group 3s, triazoles, group 7, which are the SDHIs, and group 11s, which are the QIs or strobilurins. So there's nothing we can throw at this now. So your only option now is varietal resistance in those situations. The fungicides are no longer effective. So this is not where we want to get to. Okay. Also been confirmed in WA very recently. And the issue when you get into it, it's mainly net form and net blotch. It's actually seed-borne as well. So we forget this sometimes. So you can actually get triple resistance in those other regions where it's developed. It's largely a localised problem in those paddocks. But if you get seed infection, then move that seed around, you move that fungicide resistance issue into your region as well. So it's something to be very aware of. So what's the best way to stop fungicide resistance evolving? Pretty simple. Apply no or fewer fungicides. So this is why we talk a lot in the AFREN, which we're on part of a network nationally, is start with a solid base. Start with that varietal selection. Grow as most resistant a variety as you can to the pathogens that are a problem in your region. Okay, and we look at the north, mainly thinking about stripe rust, big tick there. Where I am in northern New South Wales and up into southern Queensland, if it's more than a one fungicide spray variety for stripe rust, it'll get dumped to pretty quickly. It just doesn't fit the logistics of our system of getting across big areas, heavy clay soils. You often can't access paddocks. So big tick for us in the north. Whoops. The next one is non-chemical. So do everything else you can to stop it. So stubble management, certainly a lot of the pathogens we deal with are stubble-borne. So what do we do with that stubble? We're often restricted there because cereal stubble drives our fallow efficiency, so we've got to hang on to it. Crop rotation, we have options there. Sometimes the pathogens survive multiple years, so that makes it less effective. Good hygiene, we practice that. So at the best time to avoid or tolerate disease, we tend to be stuck with when we get sowing opportunities, and I certainly don't promote deliberately delaying sowing to avoid a disease. You compromise your potential too much. And we certainly manage the green bridge when it comes to our biotrophs, our rusts, etc., because we want to save the water on our heavy clays. So we don't let our volunteer wheat get away. And the last thing we should be doing is fungicides, only where necessary, and apply them strategically. The key things, rotate modes of action. We did some work recently. We are very much dominated by some old group 3 triazole products, so propiconazole, tebuconazole, make up anywhere from 70 to 80% of our sprays in northern New South Wales, southern Queensland. So we're exposed. Fortunately, we are seeing more mixtures come in where the triazoles are getting mixed, particularly with strobilurins. So that's good. You're forcing the pathogen to have both mutations to those different groups in one. So it's less likely and lower frequency that's going to happen. And we certainly stay within label rates. But the thing we miss with this, and I think we don't put enough emphasis on, is it all actually starts with correct diagnosis in the first place. Okay, that's the key thing to start with any management strategy, which is what I'll focus on for the next part of this presentation. But why is good diagnostics important? So this is just a map up to the end of June. New South Wales DPI with GRDC funding run a diagnostic service. Brad Baxter does it in the south. I do it out of Tamworth in the north. You can see a number there, whether they're orange is an inquiry or green is where we've got a physical sample. It's up to there, 124 actual inquiries or samples. This is the breakdown up to the end of June. 26% of what come in weren't actually disease. So the stuff in red's not disease. It was environmental. A lot of interactions with frost, droplet freeze. We had issues with insect chewing, cutting plants off just below ground. Nutrient deficiencies made up 13%. Spot-borne net blotch in barley was actually there, so we did have some disease around. Herbicide issues, and then you can see a few other diseases there, mites and other insect issue. But when you look at it, up to that end of June, 68% of what we're dealing with all come in as potential disease issues were non-disease. Okay, so it's trying to stop fungicide going out for things that aren't going to work. So why is it so important to get it right? Okay, you've got to ensure correct identification. Then you can address the actual issue with appropriate management. Fungicides won't fix non-fungal issues. So whether it's virus, bacteria, they only work against fungi. If you've got an underlying nutrient deficiency, there's not a lot of N in fungicides. They're not going to correct it, and they won't correct, frost tipping, where you just get those cold temperatures, the tip of the leaf it goes yellow. Okay, even if you use warm water in your spray, you're still not going to stop it. The other thing, it just stops that unnecessary application. So, if you put it on when you don't need it, grower doesn't see the response because it was never fungal anyway. You lose confidence from your grower. They're losing profitability. They've also just used a shot of fungicide when they didn't need it. So the supply through the season becomes an issue. And they can lock themselves out with resistance management because they've put something on there, when they didn't need it. So it changes what they can potentially use next time. So it's just important. And you're not addressing the actual underlying issue if you're getting it wrong. Certainly, if you accurately identify, it allows you to get in there early in the growing season and get on top of it. So a lot of the fungicides, when you put them on, you've got to get it early on the epidemic. You don't want to be trying to fight a bushfire once it's got away from you. Okay, before you've had that losses, you can't restore green leaf area from a disease once it's gone. And in some situations, you may be too late, and it's really hard. And I know there's a lot of psychology in decisions, but sometimes telling the grower the best thing to do is nothing is really hard. But sometimes that is actually the correct decision. Mightn't be popular, doesn't mean it's wrong. But you can use those as very strong learning opportunities as to, if you missed a timing, this is why I did advise you to spray at GS32, or you have an underlying issue here with soil type or a subsoil constraint. Let's address that first and get things right for next time. And certainly when you are getting it right, you want to know that your fungicide strategies are effective. They're doing what you expect, and I'll talk about that a bit as well. And certainly identify if you are losing, the effectiveness of some of those through fungicide resistance, and should get it tested to confirm. Certainly, keep an open mind. So I think the problem is sometimes we just jump to, it's got to be a disease because I can do something about it. So inspect, there's some logic. Where is it on the plant? Is it on all plant parts? Or dig up the roots as well. A lot of what we see above ground can be caused by issues below ground, but you have to dig them up to be able to figure that out. Note the crop growth stage, that's quite important as well. Are there distinct lesions with a border? So with our necrotrophs, you'll have a clear yellow margin, which is the toxin production, or is it just general yellowing? If it's general yellowing in the lower canopy, it's not disease. It's more indicative of some other constraint, nutritional. Other symptoms at the leaf margins with the surrounded healthy tissues, things that go to the leaf margin tend to be some herbicide interactions and other things, not classically disease. Which leaves are the symptoms on? Certainly mark those leaves to see if it's moving up the canopy, it's spreading. So most of your leaf diseases, one of the big giveaways is they start in the lower canopy. So if you're seeing stuff only on the top leaves, it just does not fit with leaf diseases distribution. And also look for those diagnostic symptoms. So off to the right, you got a picture of the fruiting bodies, pycnidia of ascochyta blight in chickpeas. So if you see those, you've got definitive evidence. So you might need to use a magnifying glass or the zoom function on your iPhone if you got one, and take some high quality photos. You can get those looked at by pathologists as well. The other big one is just use some logic. Look for patterns. So if you've got stuff where it's only really coming in from the edge, it could just be herbicide drift if you've had a spray next door. Can be pathogen movement, moving in from next door. If you've got infection on that stubble, it's not being constrained by a canopy, blowing it across. And the other ones we see is the photo down there from Kev Moore of, header dust when you harvest a crop which had ascochyta in it. That dust blows across the fence, and that's your inoculum source for next year. So you'll see clear distribution, when you've got those things going on. If it's actually quite linear and repetitive, then you really look to it being related to sowing operations. And I had a sample in this year, and bottom of the plants were going yellow, and got sent a photo of the paddock situation. You could see it was confined to a row, and it just was a blocked fertiliser tube. So that just didn't get the start of fertiliser and was suffering a lot more from nutrient deficiency. You can also see your spray applications. If something, you've had interaction there with potentially a tank mix or a reaction to a solvent or oil, you'll see it'll be in spray patterns. Got any overlaps, it'll be twice as worse there generally. And also look for wheel tracks effects. It can be compaction and interaction between things. So when they're in patterns like that, it's a bit of a giveaway. If it's individual random, that across the paddock, that could mean you've got infected seeds. So you've introduced, say, loose smut. It's just randomly across the paddock in barley. You can get airborne or rain splash where things are coming in. So you often get hot spots of stripe rust start like that. Viruses spread by aphids, they often very patchy individual spots as well. And the other big one we see, this random thing, is with varietal seed purity. We see this in Lancer wheat, which we grow a lot in my region. Very good resistant to moderate, resistant to stripe rust. But we have issues every year of stripe rust in Lancer, but it's individual plants taking a heap of rust across the whole paddock. That just shows you've got a contamination with a more susceptible variety. So do you spray a crop when the contaminant, which is actually getting rust, is less than 1% and the rest of the crop's fine? So there's some of the decisions and complications you get. The big issue with that is every year someone goes down the pub, a grower goes down the pub and says, my Lancer's full of rust, and everyone starts spraying their Lancer in that region until logic prevails. So look for the patterns. The other one, sometimes it really mimics and does look like it's disease. It can be quite uniform in patches with some variation. You know, look if there's a link back to soil type. So sometimes, particularly root diseases or other things, they interact with other constraints. Certainly stubble load with our stubble-borne pathogens, so your yellow spot, net blotch in barley, you'll usually find where you've got more stubble retention, more inoculum is retained, that's where it's worse. You can also see herbicide interactions, particularly residuals where they break down slower on certain soil types, hang around, cause problems below ground. You see that as issues above ground and interacts with some of our root disease as well. Elevation can certainly be a bit of a link to environmental things such as frost, where your cold air runs causes that issues, and certainly also interactions with root diseases can be very much linked to soil type. And in some situations, your trees are actually your friend in that picture up the top there. So when you're looking at cold interactions, frost interactions, look around trees. If it's not there, they've got some protection from the frost. It's a bit of a giveaway that it's probably something to do with that frost you had a week or 2 weeks ago. So you don't need to actually be a pathologist, but having a little bit of knowledge doesn't hurt, okay? Depending on how you use it, I should qualify. So just quickly, wheat disease. I'll just do this as an example. So we've got a range of diseases we can get in wheat, okay? We've got biotrophic on the left and necrotrophic on the right, and I'll draw out the distinction on that. So with each of them, you've got an optimum temperature range. So stripe rust cycles quickest, 12 to 20 degrees. You need 5 to 6 hours of leaf wetness. You've got an infection event. The number at the bottom is the latent periods. That's when a spore lands on a leaf to when you'll physically see pustules come out again, anywhere from 10 to 14 days. Okay, you get outside those optimum temperatures, you just extend the time it takes to cycle. Okay, some got different requirements. So look at Septoria triticide blotch, which we don't see in the north much. Likes at 15 to 20, cool as well, but got quite a long leaf wetness. So it's a function of very wet season, 24 to 48. But it's also got a very long latent period, 21 to 28 days. So it sneaks up on you. You can't see what's going on because it's hidden in that latent phase. Yellow spot, much quicker, 4 to 7 days. So it gives you a real heads up of where it's moving in the crop. The other reason why it's important when it's a biotroph or necrotroph, you tend to find some pretty significant interactions with nitrogen nutrition in particular. So your biotrophs, like higher nitrogen status crops, creates a microclimate that's actually more conducive to get that leaf wetness, humidity, etc. But also the nitrate level in the leaf is a food source for the biotrophs, so they need living cells to survive. Whereas the necrotrophs are the exact opposite, okay? So necrotroph is killing the plant cells, and it does that by producing a toxin, and that's your yellow margin is where the toxin production is occurring. And the fungus then feeds on those dead cells in behind that, and you get the tan lesion. So necrotroph, because it wants to kill cells, the less healthy the leaf is, the less N in it, it actually can spread quicker because it's got less to fight it. So N nutrition can have a lot to do with some of these. So the reason why some of this is important as well is when it comes to considering fungicides, because your fungicide curative activity is roughly half the cycle time. So if you think about a fungicide, they've got 2 modes of action. They've got a protectant, where they're stopping spores that land from initially infecting. And what they've got then is the curative activity is once a plant's in there, in the latent phase, how far back can it actually go and pick up those infections? So if you look at something like stripe rust, a 10 to 14 day cycle time, curative activity with the fungicide, and this is with your group 3s, group 7s, you don't have much curative or at all with your group 11, azoxystrobin, etc., the strobilurins. You can reach back 5 to 7 days. So any infections that happened 5 to 7 days ago, you put a fungicide on, you'll take it out without stripping any green leaf area or any pustules coming out. Once you get beyond that period, you can actually still get loose leaf area and get pustules come out. The reason that's important is when you look at something like yellow spot, really quick cycle time, you've got very, very little curative activity. So 2 to 3.5 days because it just cycles so quick. So then you think about, well, putting fungicides on, if I have got it confirmed I've got yellow spot, do I need to be in front or behind a rain event? And okay, we're used to this in ascocon and chickpeas. We know they're all got protectant activity, very poor curative. we know we have to go in front of rain events, you know, with Ascochyta blight of chickpeas. But we tend to be a bit more random in our cereals, so think about that. The other reason knowing a little bit of pathology is really important, it's very useful in preventing misdiagnosis. Okay, when you consider the latent period of these different pathogens and the rate that leaves come out. So they're both temperature linked. So a wheat crop will put out a leaf every 7 to 10 days. So therefore, you can't have infection on the top 1 to 2 leaves. A leaf can't get infected till it's out. Then you've got your latent period, say with stripe rust, 10 to 14 days. You've got 2 leaves at the top of the canopy. Until you get to GS39, when the last leaf has come out, you can't physically be there. It's not biologically possible. And we see a lot of that every year where things get sprayed, and that really basic logic just isn't applied. But there are some very good disease mimics, okay? So eliminate first other things, and disease should be your last option. And be very careful of avoiding confirmation bias. So sometimes, you know, we've all got this, the green book. We just pull that out and get flicked through, and it's just choose your own adventure. What's it going to be? Which disease is this, okay? Don't jump to that first. Just because you want it to be a disease, because you want to put a fungicide on, the grower wants to do something, he wants to put a fungus, or she wants to put a fungicide in, doesn't mean it's the right decision. So certainly cold weather does a lot. So we get this colour banding every year where we get chlorophyll production slowed. It's just where you're getting really cold temperatures overnight, then warmer days. You just get that rugby stripe, it's called. They're going to be yellow where they go right across, or you get quite red. And the distance between those spots is just the frequency of those cold temperature events. Okay, that gets confused most years. The other one we've seen a lot of is these bleached, very circular white spots in wheat and in barley, but in barley they tend to have a brown margin. It's just droplet freeze. So droplets sat on the leaf overnight, froze. It's just then how quick they get sunlight and basically thaw as to how much damage they do. So the droplet itself acts as a bit of magnifying glass, is actually just burning the leaf and causing that bleaching. And you can see a close up there, very easy to confuse with disease when you get, you know, that leaf death, but it's not tan, it's bleached. Bleach colour in cereals is very abnormal. The other thing we do a lot, and I got a Senate Classic from an agronomist the other day of, you know, just that first run of frost, you get the top leaves just tip out and go yellow. You know, when grower sends a picture to the agronomist, I'm spraying this, it's got rust in it. It's on the top leaves, can't be rust. Look around trees if it's less pronounced. So we see that most years. And actually pull out a tiller. Can be a bit of a time stamp. I don't know how well it stands out. You can see it's confined to 1 or 2 particular leaves at the tip. And the new leaf out hasn't got it. The older leaves, you know, haven't got it. Diseases start in the base. So where it is a real timestamp. We also see this, a bit of curling of the leaf, which also, again, is just related to frost. You know, it's not copper. We don't have widespread copper deficiency everywhere. And yeah. Barley, much more pronounced. And the thing that barley tricks it with is it's not just the yellow tipping. You also get these physiological spots in behind, you know, that concentrate to the tips. And, you know, everyone thinks it's spot form net blotch. So the giveaway here again is you can see it's confined to 2 leaves. There's nothing in the lower canopy, so it's not spot form net blotch because, you know, the base of the canopy wouldn't be clean. And that's really a timestamp of where the cold event occurred. It's on those 2 leaves, so you can go back in time. So fungal diseases just don't concentrate towards leaf tips. The other thing we get confused with is herbicides or nutrition. You can see contact herbicides there. We get a bit of drift, a bit more irregular, can cause lesions. Glyphosate drift, where you get that bleaching of the new growth out, and prolific tillering, we see that. And sometimes we see paraquat drift, you know, particularly where you've got some summer cropping and you might be cleaning up for that. So yeah, key thing is not disease. Look for the patterns. The other things is there are some nutrient deficiencies. So we had an issue with chloride deficiency. Everyone thought we were crazy. Graham Swanky, who's a nutrition researcher here, did some classic nutrient emission worth of glasshouse. So this just spotted up throughout the whole canopy really quickly, just in a particular durum variety, Jandroy, and it happened in really good seasons, wetter seasons. So we've got chloride at depth below 60 centimetres, but in those really wet years, the plants get lazy, the roots didn't explore, didn't hit the chloride, and they just suddenly run out of what they had, and they just spot it up, okay? So just, yeah, be a bit sceptical. The other one we see is zinc deficiency. We're all used to classic zinc deficiency where you get the big bleach, you know, the big tan lesions either side of the midrib, but sometimes we're not at chronic levels, we get more that striping. So, you know, we're pretty sure that's what it was when we tested. The other thing is there can be quite strong genetic links. So later in the season, we get melanism, or false black chaff it's called, linked to a particular stem rust resistance gene called SR2. And basically you just get humid weather and you get the overproduction of melanin, which is the brown pigment, concentrates in the glooms. Everyone thinks it must be early head blight. The easiest way to pick is look at the rachis. So that's the third picture, which is the stem in the head. With melanism, you'll see that browning is actually on the rachis as well, but you get that ladder pattern where it's only on the one side. If you flip it over, you'll have the exact reverse. You'll have that browning is on the other side. You get that ladder. Whereas with Fusarium head blight, when it's truly there, infects the stem and will take out, whole sections brown, and it's a different browning too if you look at enough of them. The other thing we see a lot of is leaf tip necrosis. Again, it's an effect of carrying particular leaf rust resistance genes, LR genes, particularly LR34 and LR46, where we run into that flowering, grain through. We get a bit of wind, hot, warm weather, and the flag leaves tip out, and everyone thinks I've got some late yellow spot or late rust. I've got to spray it. You can see there's some nice work overseas showing what the effect is there. LR34 does it worse than LR46. Fortunately, we're covered in Australia in that we've got the University of Sydney Rust Lab. They actually publish the genes in. So if you're seeing things really specific, we saw it a lot in a variety up here rated last year, LR34. You can look up the genes and go, well, that's the link. The other thing is sometimes these physiological things can be very much variety related. So we see this, what we call thumb spot, this physiological spotting. We used to see a lot in a variety called Granger, photo there from Greg Platts. I got a call yesterday and photo yesterday from Queensland. Exactly the same things happening in a newer variety, Leabrook. So it's just linked to overproduction of what they call superoxides. Happens because barley changes photosynthesis in low light situations to keep going. And when it comes out into full sites, if you had 2 or 3 days of grey weather, come out into full sun, takes a while for it to readjust its process, overproduces superoxides. They concentrate in the leaf as little brown spots, and that's what it does. And it does it in particular varieties. What about soil-borne diseases? Quickly, I'll go through that. The catch is they're a lot harder. We're used to looking at leaf disease, very visual, see a little bit, what are we going to spray? We tend to bury our heads in the sands when it comes to soil-borne disease. It's all below ground, and it's a lot harder. And we see these classic situations, whiteheads around trees and going, God, I was unlucky I got that frost. Just doesn't fit. Okay, burying your head in the sand is probably good as long as you have a look while your head's down there. The issue is with them too is because they're damaging, the root system and below ground. It's often just ill thrift, reduced tilling. They're not distinct symptoms. Often misdiagnosed as nutrient deficiency. So this is a barley crop here where grower was concerned and agronomists are concerned. The lower leaves were dying and going yellow. It's like, well, dig up some plants. They didn't have to dig them; they pulled them out. So the constraint there wasn't putting more N on it, was actually that something going on, and crown rot was actually nailing the plants right at the crown and killing the plants. They can also interact heavily with other things. So you've got one, the first photo there, so they can interact with soil type, acidity, salinity, anything that reduces the root system or makes it tougher for the roots to grow. Your root diseases are going to interact and make it even worse, even just soil type. You've got interactions with herbicide residues or drift. So the one up there was crown rot in a crop that got touched up with a bit of Roundup drift, glyphosate drift, I should say. On their own, probably would have been fine, but the combination of the two actually end up killing those plants. Waterlogging, compaction, anything that causes moisture stress and nutrition can actually make soil-borne disease a lot more pronounced as well. And yet you have to dig deeper. So this is a sample that came in from Kraken Barley back in 2022. Again, agronomist concern, lower canopies dying off. Next leaves are going yellow from the tip up. It looks classic N deficiency, that pattern, lowest leaf dead, next leaf leaving colour, next leaf tipping back, okay? But, they put a heap of N on, so it just didn't right. So have to dig deeper. Dug them up. Base of the plants were quite brown. The subcrenino on that other photo there you can see is really quite black. And what we can do as pathologists is surface sterilise and plate. It was actually common root rot, bipolaris was the problem. So it's actually restricting particularly the primary root system, just not accessing nutrition, etc., and expressing S nutrient deficiency. So you've got to get to the underlying cause. Last one is example, Exhibit A, Pythium root rot. So symptoms often distinct, confused with other things. Here's, I had a number of samples coming this year. I've got Pythium root rot. I've got yellowing the base, dead tillers, individual tillers. It's got to be Pythium. Dig it up. You can see there's actually this, shredding just below the soil surface. Pythium doesn't do that. And when they dug around, you can actually see these little beetles, African black beetle. Okay, just was never Pythium in the first place. Pythium, yet can stunt the root system, but doesn't cause that shredding. It was just the feeding of those African black beetles and their grubs that actually was causing that. So, if you don't get it right, you're not treating the primary cause. So treating the actual insects in this situation, a lot of those crop recovered, trying to drench it with metalaxyl wouldn't have done anything. Often fungicide applications are also a motive, okay? And that's because particularly in wheat where we're targeting stripe rust, it's very visual and it's that fear of stuffing up. I was going to use another word, but it was very good. And often we just don't understand the actual process and trust what we call adult plant resistance. When do you actually tell a grower, or if you are a grower, when do you actually make the decision, well, the show's over. The genetics has got this under control. I don't need to intervene anymore. Very often a hard thing to get across. So adult plant resistance, so rust is a biotroph. It needs living cells to survive. It's a cool mechanism where basically the plant senses it's got the stripe rust fungus in its cells, and it kills those cells and the surrounding cells and effectively kills the infection because there's no living cells for the biotroph to survive. The issue with that is if you've got significant infection in a crop, that adult plant resistance becomes active, then you strip a heap of leaf doing it. So all that dead tissue in behind the yellow pustules there, that's adult plant resistance killing out the infections. In situations, and you mainly see this more just in evaluation trials, if you've got heavily infected crop next to a variety that's got good resistance and adult plant resistance is active, it's still getting challenged. Worst case scenario, you hold a leaf up to the light, you'll see flecking, and that's the adult plant resistance taking out spores off that situation. This is what we advise growers and advisors to do when you get hot spots of wheat. In wheat, often you only see them because the adult plant resistance has kicked in and it's stripped out the infection and you can't miss it in your ute now. The thing to do then is, well, is this crop going to look after itself? Go to the hot spot, look at the plants just at the edge of the hot spot, hold up, look for that flecking in the leaves. That's the APR taking out the spores still come off the hot spot. Walk 10 metres away into clean crop, do it again. You'll see a lot fewer of those flecking in the leaf. You're getting exposed to fewer spores. Then that's how you know your APR is working. Now, the issue with adult plant resistance is we really need to use our fungicide strategy matched to our resistance level. And our resistance level tells us when the adult plant resistance is actually going to become active within our varieties. Sorry for anyone in the West that doesn't deal with stripe rust, but it's our biggest disease over here. So stripe rust is strange. So everything starts off at susceptible and it moves to resistance by name as it ages. And when that actual adult plant resistance kicks in is linked to the actual rating. So an MR variety, moderate resistant variety such as Raider, adult plant resistance kicks in at GS 30 to 32. Sunmaster MRMS kicks in around 37 to 39. Our Durhams, MS, anyway from Ornpeat 49 up to that, just before flowering, and MS is a later. So, we're trying to keep the top green 3 leaves green. They're our ones that intercept light, give us our yield. And our fungicides generally, only they're only protecting the leaves at the time of application, and they're only there for 2 to 3 weeks. So we're trying to time our fungicide in advance of, 2 to 3 weeks in advance of when that adult plant resistance is going to kick in. Okay, and that's why certainly we need to think also about nutrition. So with a biotroph, N can actually delay onset of APR and shifts at one disease category. You can't make a resistant variety susceptible with a heap of N, but it shifts at one. We see that every year with high N status. Flutriafol, we don't use a lot in the north, but certainly on starter fertiliser, it'll get us through to that GS32. So it's really a matter of matching when that's kicking in. And what we saw a lot last year, MRMS varieties were coming into, that Ornpeat stage. Growers and agronomists are sending pictures. Look, I've got rust, and they'll highlight. I can see active pustules. They're actually orange. They've been there a long time, and you can see all that flecking in the leaf, and the canopy is really clean. So APR is taking care of it, but convincing a grower not to go in and spray is still a challenge of understanding how it works. So walk away, it's got it covered. There's a few tips there, hopefully. So often we get, my fungicide application just didn't work as well as expected. Well, it's got to be fungicide resistance is the automatic thing. So let's just explore that in a little bit more depth. Firstly, I think we need to understand that fungicides work differently on a biotroph versus a necrotroph. So if you just look at a dodgy hand-drawn picture here with our rust, you've got a slow build-up phase, which is the little blue curve. You hit it with the fungicide, you kill the infection. Then you've got 2 to 3 weeks, With non-infection, the little red bit, then it starts to build up again. Hit it again at GS 39, kill the infection, 2 to 3 weeks protection. It slowly builds up. You get the head emergence, and even in MS varieties, you don't get a problem. So you're crashing the infection. Very different when it comes to a necrotroph, okay, because you never kill off an infection. Okay, the toxin production around the lesions there, that's killing the cells, and you can't get fungicide into dead tissue. They all move in xylem water flow. There's no water flow into that dead tissue. So what you basically get in this situation is you get a build-up, you spray it with a fungicide, you don't crash the infection, you just stop those lesions on the leaves there at the time of application from expanding any further. You reduce the production of secondary spores, conidia, on those lesions. Once that wears off in 2 to 3 weeks' time, the show's back on, okay, and it builds up really quick. Hit it a second time, you basically just stop lesions expanding, buy yourself green leaf retention for 2 to 3 weeks, then the lesions expand again and more conidia get produced, okay? So very different outcome with fungicides. Don't expect to get the same response in a necrotroph as you do in a biotroph. So my fire analogy quickly on that is, with a biotroph, a fungicide, if it's applied correctly, is more like, with a fire, you've put the fire out, totally overnight. You've got to start it up from fresh in the morning, put a heap of kindle on, let that burn for a while, put a bit bigger wood on, let that go, and then you put it on bigger bits of wood, and finally you got yourself a fire. Whereas with the fungicide application in your necrotrophs, your yellow spot, your net blotch, you've got roaring coals there. All you do is take the air off overnight. You just, fungicide wears off in 2 to 3 weeks. You pull the air on, chuck wood on, the coals just light up, and the bushfire's back on very quickly. So sometimes I think what we deal with, we think it's fungicide resistance. It's actually just our expectations aren't unrealistic, when it comes to dealing with necrotrophs. Another quick lesson we learnt, spot for net blotch 2019 around Cropper Creek. Widespread in barley crops. You can see a picture there of all the spot form net blotch, brown lesions, yellow margins. Really had low rainfall events, less than 5 mils, foggy mornings. Fair bit of barley on barley, so we're coming out of an extended drought, and we wanted a lot of stubble cover. That was an issue, but also seen at some quite long rotations being fallowed, in and out of sorghum, and we're still getting issues in that situation. And the thing that threw us a lot is there was a particular seed treatment called Sestiva, which is quite effective on the net blotches if you haven't got resistance. So it's an SDHI. It has broken down in other areas. Was just not working. So, why? The automatic assumption in this situation, we've got resistance, or we've got a dodgy batch of Sestiva. We've got to get cranky with someone. So just quickly to explain, so you've got primary infections with spot for net blotch come from the pseudothesis, those raised black structures on the stubble. So we just went out there and looked, well, where's our inoculum source? And we were finding that in there. Primary infections, 6 hours of free moisture, you've got leaf lesions. Then on those, you get secondary conidia, and you just need high humidity. So those foggy days were enough to keep it going, but they weren't enough to wet the soil. When we looked at it, some of that stubble, there's a photo from Gary Onus, Nutrinag, he picked up on it, there's actually 216 barley stubble. There's actually a moisture requirement for those structures which are inside the retained barley stubble to mature and release those acid scores. They need moisture for that. And that run of droughts, they just hadn't matured, but they coincided with 219. The other thing we looked at was, well, what was going on? Is it actually resistance? We sent samples over to Curtin University, who are the experts in fungicide resistance, 5 isolates. There was no reduced sensitivity. So we could actually rule that out. And when we looked deeper into it, it was on our heavy clay soils. We actually had the seeds sitting in dry soil, okay, for extended periods. So plants were growing on stored moisture deep, but our surface soil had just dried out. Those 5 mil foggy events weren't enough to wet that up. So with a seed treatment like Sestiva, the active's there, you need that active to come off the seed, end up into the surrounding soil, taken up the roots, then gets redistributed through the plant to control net blotch. That just wasn't happening in that situation. Okay, so it wasn't resistance, it just was a logistics and supply issue. So fungicide application disappointment in this situation doesn't necessarily mean fungicide resistance. You have to get it actually tested. So correct diagnosis in reality. So this is what we deal with a lot of situations. So this is, a typical barley sample that comes in. What is it? Ask yourself, look at that. Is it physiological spotting, spot form net blotch, both of the above? Well, basically, I've got no idea. I better contact a pathologist, which this one person fortunately did, or I've got no idea. I'm just going to spray it anyway, which is probably the norm. So what we can do as a pathologist, we can actually set up humid chambers where we take those individual lesions and we put them in an environment where we just get it to complete its life cycle. So you can see here when you break it down, the ones on the left, all those numerous small brown spots towards the tip. We didn't get anything out of those. They were just physiological. Those bigger brown spots, when we read the humid chamber, we've got conidia there of the causal fungus Pyronophora teres, which is definitive. So we can actually distinguish which is which. So that's a very quick way of doing it and getting confirmation. Yellow spot's probably our most misdiagnosed disease, but it's pretty simple. Always look for your inoculum source when it comes to yellow spot. It doesn't cause widespread yellowing. So is there wheat stubble visible in the paddock? Has it got those fruiting structures on it? And it'll be very nearby if it's causing infection. Look for distribution. So remember necrotroph, yellow margin, brown spot. It doesn't translocate the toxin. That would be stupid. And looking from left to right, there'll be more extended lesions and more lesions on the lowest leaf. Go up the next leaf, smaller, fewer, until you go up successive leaves. They haven't been out for as long, haven't had as long for expand. So look at an individual tiller. There's a clear pattern of distribution as you go up. This is the ideal situation. We had most of our yellow spot in the north last year was back where we had wheat on 2022 wheat stubble. Okay, 23 was very dry for us. We didn't break the stubble down and we actually got carry through of inoculum. And perfect photo here, you can see the fruiting bodies on the retained stubble there and the lesions in that lower canopy. So great definitive. But what we often see is more this situation where you've got a lot of general yellowing in the base. There are some yellow spot lesions there, but the big question becomes, what's the bigger issue? Particularly, we just had a rain front come through, so a lot of the debate was, well, this crop's actually chronically N deficient. It has got some yellow spot. What's the big issue? What do you want to address in front of this rain event? And often, just because fungicide's cheaper doesn't mean it's actually going to give you your better return on investment. Correct the true constraint. You can actually get a second opinion, okay, but be aware of experts. Okay, the loudest voice may not always be the corrected voice. I've certainly had a campaign up here, particularly as we get into spring and it gets quite emotive with spraying or not. I think everyone should go, all growers should go to Dan Murphy's and drink at home, because going down the pub certainly caused a lot of work for me. It keeps me in a job a lot of times, correcting some misinformation. So certainly text messages with modern technology is really good. Just take 3 high quality photos. One, what the distribution in the paddock looks like. You can see up the top there, all that yellow is a hot, pretty significant hot spot of stripe rust. Picture of a whole plant, ideally washed out, so I can, pathologists can zoom in and have a look at the distribution of the plant, what's going on the roots, and a close-up of the actual symptoms in particular what you're worried about. Definitely include your name, variety, growth stage, all good information, rough location where it is, brief message around your concern and other relevant information, okay? Don't just send it blind. We also welcome agronomist reports of things you're seeing or stuff you've learned or dealing with your growers, all good information for industry so we can tailor our messaging. Certainly if you're submitting samples, particularly with root diseases, 2 to 10 symptomatic whole plants, knock off the soil. Don't wash them out, but just don't leave the soil. It costs too much. If you do wash them out, wrap them up a bit like a bouquet. So basically wet paper toweling just around the root mass that's washed out. Then put a plastic bag around that, but rolled right down and just sticky tape that. It'll keep it fresh when we get here. And certainly best if you send a good and a bad sample, because what we're really looking at is difference between visual differences, what's going on with the root systems causing the problem there. And we do send a fair few off for quantitative PCR analysis, looking at which actual pathogens are different within the root systems themselves as a in crop diagnostic. Don't send fully inside a plastic bag. By the time that gets here, they'll look like the picture on the top right there. They're just gone mouldy. Can't do anything with it. Wrapping them in newspaper is really good. And the details there, perfect one there that come in the other day. Happened to be a vetch sample inside. It's all nice and fresh. So, and certainly if you're only concerned about particular leaf diseases, just send a message of where it is in the crop. You can send it in an envelope, small envelope, 10 to 20 symptomatic leaves. And we can certainly do humid chambers on that and confirm and send those then off for fungicide resistance testing if we need to. Certainly, again, include your name, mobile number so we can get back to you with the information, variety, growth stage, etc. Yes, don't assume that you're the only one sent it in. We do get a number of samples every year with no name, no nothing. So that's difficult. So you'll have your own key contacts in your own states, but for New South Wales, certainly at Wagga Wagga, Brad Baxter for winter cereals, Kurt Lindbeck for canola and pulses, myself here up in northern New South Wales, and I do push into southern Queensland a bit for winter cereals and I guess, general things if you want me to find experts on others. Very colourful too. This was a sample that come in wrapped in newspaper, which I happened to feature in. Someone thought it'd be quite funny to, put a bit of graffiti on. So yes, all contributions welcome. Zaretza Jurek is our field crop entomologist if you've got insect issues, and Job Van Loor if you've got broadleaf virology or faba bean specific issues. So they're our people here. And just to finish off finally, if you want more information, particularly on canola, pulses, etc., I'm very much cereals. I stayed within my safe zone. We have got some workshops coming up funded through GRDC at Griffith on the 12th and 13th of August, and Wagga Wagga will actually go through this in a lot more detail, what to look for, how to distinguish a crop across all those different crop species. So thank you very much.
Excellent. Thank you, Stephen, for that. That was really informative and also really entertaining. So I hope everybody enjoyed that. If you have any questions, please put them in the Q&A box at the bottom of your Zoom screen there, and I can ask those of Stephen, and hopefully he'll be able to answer those for you. I'd like to also reiterate that's really good information, Stephen, that you gave to people about how to send their samples and how to include pictures and information for the pathologist. That's really good. I want to also add to that, that'd be really good if you want to get fungicide resistance testing to also add what your fungicide regime would be, so that we can have a look and see whether those find any mutations here in the lab, whether they actually marry up with what's happening in the field as well. So thank you very much for that.
Yeah, and that can be quite good information too, also for just diagnosis. I had a spot form net. Well, I was convinced it was spot form net blotch. Distribution's right, the lesions look right. Did a humid chamber, and just not getting any canidia out of it. Yeah, it was sprayed. Day before they sent the samples. So
yeah, it's interesting. So many things to think about.
Yeah.
Excellent that we have the experts like you there helping us all out. That's great. Well, it looks like there are no questions coming in at the moment. So hopefully Steve has been able to answer all the questions that you already had in your mind by coming along to our webinar. So thank you all, everybody, for attending the AFREN webinar. Look out for more of our webinars as they're coming up over July and August. And if there's no more questions, I'd like to thank you on behalf of, do you have something?
Oh, no, I was just going to say, just if anyone does have specific questions, my mobile, hopefully you grabbed it. If not, go back. Yeah, just send us inquiries over the next couple of days or give us a ring. Not an issue at all.
And if you can't get in touch with Stephen, I'm quite happy for you to send any email notifications to AFREN. That's afren@curtin.edu.au, I believe. Or you can look at our website, afren.com.au, where we have all the details as well. So thank you all on behalf of Stephen and myself and everyone in the AFREN network for attending our webinar today. And once again, if I could ask you to spare, hang on, I have a question here. Just one sec. So, Stephen, can you say, from Lockie Wilson, for Septoria avenae blotch, are the conditions for infection the same as triticide blotch?
Yeah, largely in terms of temperature. I'd have to look up, I'll get you, if you get me to text you, I'll look up exactly what it is. But yeah, it's a different, obviously, it's a different species in the same group, but yeah, it's generally Septoria's got longer latent periods, but I'll double check that and the exact temperatures. So sorry, I don't know that off the top of my head, but I can certainly find the info.
That's right.
We don't. Yeah, but interesting. You raise that. We don't see a lot of that. Tend to see a lot more of leaf blotchy notes where it's Porinophora that's actually doing it. So yeah, it can be good to just get it confirmed as well. We have had a number where we thought that it's either Septoria or leaf blotch, Porinophora innates this year, but it's actually just some waterlogging. We've been quite wet, so you get.
Oh, we seem to have lost Stephen there. Always the way when you're wanting to get an answer. All right, we might have to leave it there, Lockie. Sorry that Stephen couldn't finish your answer, but please do get in contact with him, or you quite I'm quite happy for me to pass any information on as well if you want to get in contact with me. Sorry, Stephen, you're back. We lost you there halfway through your answer.
Yeah, no, just other things can, just with that, good to get it just confirmed. So I'll reach out and we can get a sample looked at to check that's what it is as well.
Excellent. Great. Thanks for your question there, Lockie. So we might leave it there. Thank you all very much for attending today for our webinar. On behalf of Stephen and the AFREN Network, thank you to everyone. And if you can spare a small amount of time to finish our survey at the end of this webinar, we would really appreciate that to help us do better. Thank you so much.